Protein synthesis is the making of protein. It has two parts, transcription and translation. Transcription is making an RNA copy of a gene of DNA in the nucleus and exporting that RNA copy to the cytoplasm. Translation is having a ribosome use mRNA to make a protein. Proteins are one of the 4 macromolecules necessary for life and does a wide variety of functions such as being an enzyme, being involved in transportation, and providing structure. DNA is information that codes for a protein. DNA is one of the 4 macromolecules essential for life and is made only with the 6 basic elements used by life. DNA is an abbreviation for deoxyribonucleic acid. DNA is composed of a sugar phosphate backbone and a nitrogenous base. The sugar backbone is made of a phosphate group, deoxyribose, a sugar. The deoxyribose is the first part of DNA's name. DNA is a polymer, a long chain of molecules, with its monomer called nucleotides. When nucleotides bond together, they form a double helix. DNA bonds together through its backbone, or with the other side of the helix through hydrogen bonds between the nitrogenous bases. When DNA backbones bond together, they end in 3' or 5', which was reversed in the opposite strand as shown in this image. The 5' end has the phosphate group, where the 3' end has the hydroxyl group. Nitrogenous bases are either A, adenine, T, thymine, G, guanine, or C, cytosine. Only A bonds with T and G bonding with C because of where the hydrogen is in the molecule, with an exception of some errors. This rule is called Chargoff's rule. Eukaryotic cells have DNA in the nucleus stored in semi-condensed chromatin shapes, meaning they are compacted, but not as much as they are in mitosis. Chromatin proteins such as histones compact and organize DNA into these condensed forms. These compacting proteins guide the interaction between DNA and other proteins, helping control which parts of the DNA are transcribed. mRNA is short for messenger RNA, or ribonucleic acid. RNA differs from DNA by having a ribose instead of a deoxyribose, by only having a single strand, and by having eutamine U instead of T. mRNA is made during transcription. DNA that codes for mRNA are in sections of protein-coding DNA called genes, which range from a few hundred DNA bases to more than 2 million bases long. mRNA should only be as long as its corresponding gene. An mRNA vaccine is a vaccine that uses mRNA to build a viral protein, specifically antigens, molecules that bind to an antibody, that stimulate the immune system. The only approved vaccines of this kind are for COVID-19 and produce a spike protein used on the outside of the virus that is used by the virus to enter a host cell. Mrna's vaccine might be a predecessor to these vaccines. A gene starts shortly after a promoter, a sequence of DNA that tells a protein to start transcribing a gene. These promoters are non-protein coding sections of DNA and provide a binding site for that transcription protein. Non-coding sections of our DNA make up most of our DNA. Other non-protein coding DNA sections include enhancers, silencers, insulators, tRNA code, and rRNA code. Promoters provide binding site for proteins that help activate transcription. Enhancers get bound to specific transcription factors, which are proteins, that cause looping in the DNA to get closer to the regulated promoter and can stimulate the transcription protein to stimulate transcription. Hormones, also protein, bind to intercellular regions such as in the specific transcription factors and stimulate transcription. Silencers are sections of DNA that are binding sites for specific transcription factors that silence or inhibit transcription and are found distant from genes in both directions of the gene. Silencers work the same as enhancers, but with the opposite effect, inhibiting rather than stimulating. Insulators provide binding sites for proteins that control transcription in many ways, including by preventing enhancers from enhancing, known as enhancer blocking insulators, and or by preventing structural changes in the DNA that repress gene activity, known as barrier insulators. tRNA, transcription RNA, and rRNA, ribosomal RNA, are involved in translation and will be talked about later. Introns are sections of DNA in a protein-coding gene that get cut out of the mRNA and can have regulatory DNA within. Non-coding regions of DNA have not yet been fully understood. One cause of the difference between different types of cells are different gene regulations, patterns that turn on and off genes. Gene regulation is an important part of development. Gene regulation makes only a portion of genes active at any time in your cells and usually affects gene expression during transcription. Signals from the environment or other cells activate proteins called transcription factors, which bind to regulatory regions of a gene and increase or decrease the level of transcription. By controlling the level of transcription, this process can determine when and how much protein is made by a gene. Transcription starts with RNA polymerase, the transcription protein, and is initiated by a promoter. RNAP and RNA pol is short for RNA polymerase. Promoters containing specific DNA sequences such as response elements provide a secure initial binding site for RNA polymerase and for proteins called transcription factors that recruit RNA polymerase. Response elements are short sequences of DNA with a gene promoter or enhancing region that are able to bind specific transcription factors and regulate transcription of genes. A transcription factor, or a sequence-specific DNA binding factor, is a protein that controls the rate of transcription of genetic information from DNA to messenger RNA by binding to specific DNA sequences. The function of transcription factors is to regulate, to turn on and off genes in order to make sure that they are expressed in the right amount, in the right cells at the right time, throughout the life of the cell and organism. These transcription factors have specific activator or repressor sequences that attach to specific promoters and regulate gene expression. In prokaryotes, the promoter is recognized by RNA holoenzyme, specifically the sigma subunit, which in turn are often brought to the promoter DNA by a nearby activator protein. In eukaryotes, at least 7 different factors are necessary for the binding of an RNA polymerase II to the promoter. RNAP2 is responsible for transcribing mRNA, microRNA, and most small nuclear RNA, snRNA. snRNA is involved in splicing. Only in eukaryotes are other RNAPs, as identified by the Roman numerals 1, 2, and 3, that transcribe different RNA, and RNAP 4 and 5 are only found in plants. Eukaryotes also need transcription factors to help the RNAP. Prokaryotes use a specific RNAP called RNA polymerase pol holoenzyme in transcription. This enzyme is made of a core enzyme which has primary parts of 2 alpha units, 2 beta units, beta and beta prime, and an omega unit, which reads and builds the RNA. This is simpler than the core enzymes in eukaryotes. Its other part binds to the promoter region and is called a sigma subunit or sigma factor. This holoenzyme makes all the different kinds of RNA in prokaryotes. When something is upstream, opposite the direction of transcription, it is said to have a negative number element, where the number is the base pair distance from the gene. In prokaryotes, the usual promoters are 35 and 10 nucleotides upstream, with 7 base pairs in between. But more promoters can be used. The negative 10 promoter is called the primno box. These further upstream promoters are a blurred line between promoters and enhancers. Prokaryotes use the specific nucleotides in each of these promoters to determine which direction is downstream. One way to distinguish the promoters is by the 3 hydrogen bonds in C and G nitrogen bases represented in the negative 35 promoter that aren't found in the negative 10 promoter. Scientists are not sure how RNAP gets to promoters and avoids wasting time binding to non-promoter sections of DNA, but scientists think it is caused by faceted diffusion. According to my PNAS.org source, they tested using bacterial transcription in the well-studied Escherichia coli RNAP, where the RNAP reaches a promoter by faceted diffusion, FD. Binding to a non-promoter DNA was shown to be much slower than binding to a promoter containing DNA of the same length, indicating that non-specific bindings are not on the pathway to promoter bindings. This data supports a direct binding mechanism where the RNAP reaches the local vicinity of promoters via 3D diffusion, and that binding may be accelerated by atypical structures or dynamic features of promoter DNA. 3D diffusion models, though, show that the RNAP binds to the promoters to the limit or exceeding the limit of the models. Bacterial RNAP holoenzymes have multiple DNA binding domains that interact with different parts of the promoter sequence, with some being flexibly tethered. This flexible tethering makes possible the relative movement of the DNA binding domains over a few tens of base pairs, a distance similar to the promoter. Because sigma 54 RNAP has the advantage over sigma 70 RNAP that promoter recognition may be more specific, further tests use that RNAP variant. Binding of RNAP to the promoter DNA was more frequent, and about 90% of the events observed on the promoter DNA are promoter-specific binding events. The observed binding to non-promoter DNA mostly represents generic sequence non-specific binding of the polymerase along the DNA. The data suggests that the rate of promoter-specific binding is independent from the length of flank DNA. The binding of sigma 54 RNAP R to the promoter P initially forms a kinetically unstable closed complex, RP1, which then isomerizes into a much more kinetically stable closed complex, RP2. RP2 is the precursor to open complex formation and subsequent RNA synthesis. There are more sequence nonspecific complexes that form more than 7 base pairs away from the promoter that significantly accelerate promoter binding by sigma 54 RNAP. Faceted diffusion over distances longer than around 10 base pairs is not involved in promoter bindings. More non-promoter DNA means more short-lived, non-specific bindings and is unrelated to the promoter binding rate. It is very rare for long-distance sliding to create promoter bindings. Non-promoter DNA does not accelerate promoter binding by sigma 54 RNAP. RNAP might know how to swim to a promoter in addition to getting help from other proteins to get motion. One-dimensional sliding on DNA is not used to a major degree to find promoters in some environments or some RNAPs. After the RNA polymerase holoenzyme binds to the promoter and makes an RP2 complex, it starts building the mRNA strand. In prokaryotes, the holoenzyme core units release away from the sigma subunit and move downstream. In eukaryotes, the RNA polymerase II does not attach directly to promoter like bacteria. Instead, helper proteins called basal or general transcription factors bind to the promoter first, helping the RNAP attach to the DNA. Transcription factors used by eukaryotic RNAPs are called general transcription factors. Many eukaryotic promoters have a sequence called a TATA box or TATA box, which plays a role much like that of the negative 10 element in bacteria. This TATA box is first recognized by one of the general transcription factors. Then other general transcription factors and eventually RNA polymerase II bind to this TATA box and other transcription factors. Eukaryotes have further upstream a CAC box and a CG box as promoters. The TATA box contains lots of A's and T's, 2 hydrogen bond nucleotides, which make it easy to pull the strands of DNA apart due to decreased hydrogen bonds needed to start unzipping. After binding, RNAP quickly separates the DNA in the elongation part of transcription and reads from 3' to 5' the template strand. Elongation is the second part of transcription. Elongation is the same in prokaryotes and eukaryotes. In elongation, RNAP is like all the enzymes in DNA replication put together. RNAP also keeps the DNA apart while replication occurs, stabilizes the DNA, and straightens or unwinds the DNA. Scientists don't know the involvement of RNAP in proofreading, though. While reading the DNA, RNAP builds RNA that is a copy of the coding strand, the strand opposite the one being read, which is achieved due to complementary base pairs of RNA matching the template strand by following Chargoff's rule. RNAP doesn't build in the 5' to 3' direction, only in the 3' to 5' direction. RNAP always knows which strand to read because DNA and RNA can only be built in the 5' to 3' direction. The first letter that RNAP transcribes is the +1 site or the transcription start site, the first nucleotide in a gene which is downstream the -1 element by 1 nucleotide. Transcription ends with the third stage, termination. Termination happens when a terminator section of DNA is read, which causes the transcribed RNA to be released from RNAP. One example of this is the hairpin mechanism. In prokaryotes, termination is mostly row-dependent or row-independent. In row-dependent termination, the RNA contains a binding site for the protein called row factor. The row factor binds to the sequence and starts moving up the mRNA towards the RNA polymerase. When it reaches the polymerase at the transcription bubble, row separates the RNA transcription and the template DNA strain apart and release the RNA molecule, ending transcription. Another sequence found later in the DNA, called the transcription stop points, causes RNA polymerase to pause and thus help row catch up. Row-independent termination happens during specific sequences in the RNA template strand called inverted repeats. The RNAP hits a region rich in C and G nucleotides, and the resulting DNA wants to, and does, fold back on itself to bond the complementary C and G nucleotides together. What the nucleotides at the curved part of the RNA doesn't matter. The result is a stable hairpin that causes the polymerase to stall. The hairpin is followed by a series of U nucleotides in the RNA binding with the complementary A nucleotides in the DNA, which are said to be a weak bond. This, coupled with the stalled polymerase, produces enough instability for the enzyme to fall off and liberate the new RNA transcription. The hairpin loop also attracts a lot of cleavage enzymes that separate the RNAP from the RNA. In eukaryotes, one termination for mRNA that scientists don't fully understand begin when a polyadenylation signal appears in the template strand that the RNAP reads. This sequence is AAUAAA, or AAUAAA. Then the RNAP, or other enzymes, cut the RNA, which release it. Then the RNAP, strangely, transcribes 50 to 2,000 more nucleotides that seem to be a waste byproduct, before it eventually detaches through not fully understood mechanisms. In eukaryotes only, RNA molecules must be processed after transcription. They are spliced and protected. This is called post-transcriptional RNA modification. Prokaryotes and lower eukaryotes lack introns needed for splicing, with some exceptions. Bacterial introns include the group 1 and group 2 introns. Even viruses and archaea have introns. Prokaryotes don't need mRNA to be transported out of the nucleus and don't need protection. This makes transportation happen much faster in prokaryotes than eukaryotes after translation. For eukaryotes, processing has its second step being splicing by a complex of enzymes called a spliceosome, and leave behind the rest of RNA called exons. This spliceosome can also be called SNRPs, or short nucleotide regulated proteins. After introns are cut out, the exons are fused back together. In alternative splicing, the mRNA is spliced differently, resulting in different proteins or variations of a protein. This affects introns, but not exons. In this image, exon 3 and 4 can be mistaken for introns. The intron containing mRNA is nicknamed pre-mRNA, or heterogeneous nuclear RNA, hnRNA, which becomes primary mRNA or mature mRNA after the spliceosome is used. RNA splicing begins with assembly of helper proteins at the ends of introns, bringing the ends together. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. One end of the intron is cut and folded back on itself, which forms a loop. The spliceosome then cuts the RNA to release the loop and joins the two exons together. The edited RNA and introns are released and the spliceosome disassembles. The ends of the introns are, usually, going from 3' to prime, GU and AG. In the middle of the introns is an A nucleotide that has a specific OH group that binds to the GU group on the 3' end to form a loop. SMA, spinal muscle atrophy, is one example where splicing is done incorrectly, resulting in a deficiency in SMN protein. Beta thalassemia is caused by a protein with an intron not removed. Before splicing is protection, where mRNA is protected from things outside of the nucleus while it's in transportation. Here, mRNA gets a 5' cap and a poly-A tail put on their ends. The 5' cap is turning a triphosphate group into two phosphate groups using RNA triphosphatase. Then a guanosine monophosphate GMP molecule is added, which started as a GTP but had two phosphates removed during the implementation. Guanylyl transferase is what added this GMP. Then SAM, adenosylmethionine, which adds a methyl group, CH3, to the 5' cap. Together, these added molecules are 7-methylguanidine group, 7-Mg group. This capping is used to initiate transcription and acts as a signal to allow entrance into transcription. This cap also prevents degradation from nuclease enzymes. Poly-A polymerase adds a poly-A tail, or a bunch of up to 200 adenosine nucleotides to the 3' end. These ends play a role in transportation. Errors in RNA are not carefully monitored in transcription because RNA is temporary. The areas immediately before and after the mRNA do not get translated and are called the 5' and 3' UTR, untranslated regions. In RNA editing, some cells have an enzyme, such as cytosine deaminase, where a letter in an RNA strand is changed from a CAA to a UAA with this enzyme. When UAA is read, it is a stop codon, which shortens the resulting protein. In prokaryotic cells, translation can happen while the mRNA is being transcribed. When multiple translations occur on a strand, when about one ribosome attaches to an mRNA for every 10 nucleotides, the mRNA and ribosomes become a polyribosome. Ribosomes are an organelle in the cytoplasm of the cell that use mRNA to build proteins. After mRNA is being made, it is sent out of the nucleus and into the cytoplasm, where it finds a ribosome. Before I talk about translation and how mRNA needs to travel in eukaryotic cells, I want to talk about RNA and tRNA and how and how it is transcribed and shaped. rRNA stands for ribosomal RNA, and they make up ribosomes. They are transcribed by RNAP1. tRNA stands for transfer RNA. tRNA is partially formed by hairpin loops. It is transcribed by RNAP3. RNAP3 transcribes other RNAs besides tRNA, including snRNA. tRNA has a specific shape for each amino acid. The amino acid on a tRNA will match the one mRNA codes for, and the tRNA will attach it to the growing polypeptide chain. The specific shape of a tRNA comes from the nucleotides naturally bonding together on their letters. This is why it looks like a complex double helix and partial hairpin loops combined. A protein is made of these amino acids. The bonds between amino acids are peptide bonds. tRNA can also be spliced, resulting in tRNA-derived fragments, or TRFs. The 3' loop, or as shown in this yeast tRNA, the right loop, is called the T-arm, and allows for the RNA to enter the ribosome. The bottom loop contains the anticodon. The variable domain varies between tRNA and can be further researched by someone else for its importance. The amino acid in each tRNA is attached to the 3' end, the OH end, of the tRNA via a covalent bond. This 3' end usually has the letters CCA and is like a little cup, and is the CCA domain. rRNA in orange and ribosomal proteins in blue for a ribosome subunit. A ribosome is made of multiple subunits of different rRNA, the small subunit and the large subunit. I assume RNA coils up based on its base pairs. In eukaryotes, the large subunit or large ribosomal subunit can be described by a Van Berg unit where it is a 60S ribosomal subunit. In eukaryotes, the small subunit is 40S. Together, these subunits make 80S on the Van Berg unit system. In prokaryotes, the large and small subunits are 50S and 30S respectively, while together they are 70S. In prokaryotes, antibiotics can target and inhibit these subunits. Antibodies include aminoglycines such as gentamicin and tetracyclines such as doxycycline, tetracycline, and minocycline that inhibit the 30S subunit. Macrolides such as azithromycin, clarithromycin, and azermocyan target and inhibit the 50S ribosomal subunit. Because of stem loop configurations, rRNA can form tight and specific interactions with ribosomal proteins to form ribosomal subunits. These ribosomal subunits contain basic residues, as in pH, and aromatic residues, such as phenylalanine, trypsin, and tryptophan, allowing them to form chemical interactions with their associated RNA regions, such as stacking interactions. Ribosomal proteins can also cross-link to the sugar phosphate backbone of rRNA with binding sites that consist of basic residues, i.e., lysine, and arginine. All ribosomal proteins, including the specific sequences that bind to RNA, have been identified. These interactions, along with association of the small and large ribosomal subunits, result in a functioning ribosome capable of synthesizing protein. rRNA types being used change between the large and small subunit. While the small subunit in eukaryotes is made of a small rRNA of around 180 nucleotides, the large subunit in eukaryotes has two small rRNAs and one large rRNA of around 5,000 nucleotides. Eukaryotic rRNA has over 70 ribosomal proteins which interact to form larger and more polymorphic ribosomal subunits than prokaryotes. rRNA can interact with codons to restructure the entire ribosome. Ribosome assembly mostly occurs in the nucleus. There can be several million ribosomes in a typical eukaryotic cell. After any RNA in eukaryotes is made that needs to go to the cytoplasm, it has to go through a nuclear pore, which is larger than the condensed RNA. This is because nuclear pore complexes are massive. In order to pass through the double plasma membrane, it gets transported by transport proteins. Once these transport proteins combine with the mRNA, it is called an MMP, which moves by using beacons and Brownian diffusion. This also makes mRNA exit the nucleus evenly. Although mRNP complexes move by diffusion, their mobility is curtailed upon depletion of ATP from cells. The solid-state active transport hypothesis and gene gating hypothesis tried to fix the problem that interphase nuclei are so vicious that large mRNP particles will not be able to diffuse freely with them. Another hypothesis is that the interphase DNA has sections of high density and cytoplasmic-like density called interchromatic spaces. After a mRNA finds a ribosome randomly, translation occurs. This is when mRNA codes for a protein using tRNA and ribosomes to make a protein. It is when mRNA is translated into an amino acid chain. Ribosomes have 3 binding sites where tRNA can dock: the A site, amino acyl first position; the P site, peptidyl second position; and the E site, the exit position. When tRNA enters the ribosome with an amino acid, it gets checked to see if it matches the mRNA code on the A site. At DNA, 3 letters of code are grouped together into triplets. This group isn't physically spaced, just read with this distinction. When the mRNA is transcribed, it becomes a codon. When mRNA gets read, it is read each codon at a time by a tRNA. The bottom 3 letters of tRNA that match to the codon is called an anticodon and use Chargoff's rule, complementary base pairs, to match with the mRNA. When mRNA gets read by a tRNA, the tRNA enters to the A site. When the correct tRNA enters, it matches an amino acid to the polypeptide chain with a twist and moves to the P site, where it waits for another tRNA to move it to the E site. There are 20 amino acids that organisms use, 9 of which need to be eaten by humans because they're not self-produced by us. More amino acids could be used, but are not. This means that there are at least 20 tRNAs. As you can see here, the only difference between each amino acid is below the first row, the R group or side chain. Here is a chart of abbreviations for each amino acid. To match these tRNA amino acids with each codon, we use a codon chart. In this chart, you read the left side, then the top side, and you highlight the box that is intersected by each column. Then you read the third base of the codon, and you look at the left column and see the 4 letters on the right side in that left column. The letter in that right column, that is the third base, is then intersected with your highlighted box to get an intersection that is the amino acid for that codon. I prefer the circular codon chart where you read it by going outwards. One way to do this is by starting with DNA. First, you change the DNA to RNA by switching the base pairs according to Chargoff's rule, but with U instead of T. Then you go outwards on this chart per each letter to get each amino acid, and you repeat this for every codon to get the entire protein. A law of genetic code is that DNA is commaless, meaning that there are no letters between each codon. Each codon is read back to back without base pairs between each codon. The exception is with viruses. Genetic code is also non-overlapping, meaning the letters of one codon are not in another codon, with an exception in viruses. Genetic coding is also redundant or degenerate, meaning not all codons have their own tRNA or amino acids, which give room for errors. The exception to this law is that methionine and tryptophan only have one codon, AUG and UUG respectively. An easy way to find the total possible codons is 4 to the power of 3, which equals 64 total codon permutations with repetition. One thing you might notice is the 3 stop codons, which tell the ribosome to stop translating. Here is the amino-acyl tRNA synthase for methionine that uses ATP to bond methionine to a tRNA. This tRNA can fit properly into this specific tRNA synthesis and has the correct anticodon. The first step is to attach ATP to the amino acid to make a pyrophosphate. The amino acid attaches to the AMP called Acyl AMP. Then the tRNA is identified and combined with the amino acid, which releases the AMP. The first step is to add ATP to the amino acid to make a pyrophosphate. The amino acid is attached to AMP, called aminoacyl AMP. Then the tRNA is identified and combined with the amino acid, which releases the AMP. The left loop of tRNA is the DR, and allows for identification by this enzyme aminoacyl tRNA synthase. When tRNA has its amino acid, it is called a charged tRNA. Some tRNAs can wobble or bind to more than one base pair, allowing for fewer tRNAs to be coded. Wobbling rules include that a G in the anticodon can pair with a C or U, but not an A or G, in the third position of a codon. Wobbling rules include that a G in the anticodon can pair with a C or U, but not an A or G, in the third position of the codon. Wobbling is the cause for the redundancy law. Wobbling, for example, in AUA, AUU, and AUC can be explained by I design. which is complementary to each of these three letters and is on the end of the tRNA anticodon. The wobble effect also decreases the risk of mutation by reducing the effect of a mutation in mRNA code. The first stage of transcription is initiation. During initiation, an mRNA strand forms a loop, and the small ribosome unit, the bottom of the ribosome, hooks onto it. The small subunit then finds a sequence of bases that signal it to begin transcription, called the start codon, AUG. The process of this codon and tRNA relationship explains why wobbling should not occur with this tRNA. Finally, a tRNA with UAC anticodon pairs with this start codon and takes up the second position, P site, of the ribosome. This tRNA carries the amino acid methionine, or MET. At this point, the large ribosome subunit gets in position above the mRNA and small subunit. In eukaryotes, they don't have a Shrine-Digaro sequence, just a start codon. Here, a eukaryotic initiation factor binds to the 5' end and identifies it, as in sees its existence. It is specifically called eukaryotic initiation factor type 4. Then some initiation factors help the small subunit bind to the start codon. Then eukaryotic transcription factor type 2 helps bind the tRNA for methionine rather than N-formal methionine to the start codon. Then the GTP, large subunit, and release of eukaryotic initiation factors work the same way as in prokaryotes and eukaryotes, with some slight differences being in the name of specific factors. Here, the assembled ribosome starts to slide along the mRNA. A tRNA molecule will bind to the first position site, the A site, of the ribosome, and its amino acid forms a polypeptide bond, with methionine. This tRNA enters with the help of an elongation factor, eukaryotic elongation factor type 1. On the EEF1 is a GTP that has the energy to enter the ribosome, and this factor gets spit out after the tRNA enters. Amino acids come together because a nitrogen on one amino acid attacks the amino acid's N-terminus attacks the other amino acid's carboxy terminus, or carboxyl group. The ribosome has an enzyme called peptidyl transferase that helps or catalyzes this process of combining amino acids. After the attack, the tRNA in the A site has the polypeptide chain. Afterwards, this tRNA moves to the second position, and the tRNA for the AUG codon enters the E site. The moving of the mRNAs and attached tRNAs here is called translocation. Translocation is done by the protein eukaryotic translation factor type 2, which uses GTP energy to do the shifting. Now the first position site is ready to accept a new tRNA. The tRNA in the E site exits the ribosomes and gets to recharge and enter the A site again. This process keeps going until the ribosome gets to a stop codon, but with more amino acids per each cycle. Here are some images of termination that I don't understand. Termination is the third and last step in translation. When a UGA, UAA, or UAG codon enters the A site, no tRNA reads it. Rather, a release factor comes in. This factor binds to the stop codon and stops the translation process. It cleaves the polypeptide chain from the tRNA in the P site. What changes when the ribosomes are membrane-bound on the endoplasmic reticulum, ER, rather than these free ribosomes? This only occurs in eukaryotes due to only them having membrane-bound organelles. Proteins that are made in rough ER tend to be for when the protein is to be secreted from the cell, incorporated or embedded into the cell membrane or organelle membranes, or become part of a lysosome. When a free ribosome makes a polypeptide chain, the first amino acid could have a signal sequence that is recognized by a protein, signal sequence protein or particle, SRP. Once it binds to the amino acid, it has a high affinity for receptors on the rough ER and drags it to the rough ER membrane. These receptors are signal recognition particle receptors and attached to SRP. Also on the rough ER membrane is the Translocation protein, which is started in the closed state. SRP and SRP receptors both have a single GTP attached to them, which gets broken down when the SRP and SRP receptors bond together. When these two GTPs get broken down, the translocation opens and the ribosome lines up perfectly for the amino acid chain to go down the translocation into the lumen of the rough ER. The SRP gets spit back out to find another amino acid chain before the amino acid goes through the translocation. Now that the signal sequence was used, it is unnecessary and gets cut off by a signal peptide base within the rough ER, where the amino acids will degrade into separate amino acids by other enzymes. When the stop codon is reached, the polypeptide chain is finished and enters the rough ER, where the translocation closes and the ribosome disassociates. Non-rough ER proteins from free ribosomes include cytosolic proteins used in metabolic reactions and glycolysis, nuclear proteins involved in transcription, replication and modification, mitochondrial enzymes used in mitochondrial processes and certain metabolic processes involved in the mitochondria, and peroximal enzymes used as catalysts and are involved in peroxisomes. So now we have our polypeptide chain. The nucleic acids are chained together to make the correct protein, now go through protein folding and post-transcriptional modification to get their shape. In post-transcriptional modifications, things are either cut off or added. Some examples of post-transcriptional modification are adding a sugar residue in glycosylation, such as for antigens and transportation, adding a lipid for lipidation, such as for cells and organelle membrane proteins, adding a phosphate group in phosphorylation, used in protein kinase A, PCK, and cyclin-dependent kinases, CDK, where the phosphate group can activate or inhibit proteins. in adding a hydroxyl group in hydroxylation, adding a hydroxyl group in hydroxylation used in collagen, adding a methanol group in methylation to decrease transcription, and adding an acetyl group in acetylation to increase transcription. Modifications include cutting amino acids and many other modifications. Trimming can be in digestion to make inactive tryptogen or active trypsin. Places to do further research include tRNA transcription, rRNA transcription, enhancers, insulators, silencers, miRNA, incRNAs, telomeres, satellite DNA, snRNA, siRNA, which are short interfering RNA that can interfere with mRNA, intergene regions, why E. coli is the preferred species for prokaryote research, and why eukaryotes is preferred species for and why yeast is the preferred species for eukaryotic research, etc. Look in the description for my list of sources, and look in the pinned comment to get the full transcription, to get the full script.